This question is from IIT JAM, and in this video we will going to learn some basics of PCR. Then we will try to solve this question. PCR is used to amplify the number of DNA molecule or simply increase the number of DNA molecule. In PCR, first we raise temperature up to 94 to 96 degrees Celsius, and at this temperature hydrogen bonds between two complementary DNA strands are break and two complementary DNA strands are separate from each other. This step is known as denaturation. In PCR, we use primers. These primers are 19 to 20 nucleotide long approximately, and they are complementary to the template molecule. In second step, we reduce temperature up to 55 degrees Celsius, and at that temperature, this primer attach complementary to the template stand. Here you can see two primers, but in PCR we use so many copies of these two primers. This step is known as annealing, and after that we raise temperature up to 72 degrees Celsius. And in PCR, Taq polymers and DNTPs are also used. At 72 degrees Celsius, Taq polymers start adding nucleotides on 3' end of primer, and these nucleotides are complementary to that of template stand. You can see in this video, we perform PCR with one DNA molecule, and after one cycle we get two DNA molecules. And after second cycle we get total 4 number of DNA molecules. Then you can say after every cycle the number of DNA molecules is double. Then get back to the IIT JAM question. While performing PCR, a student forgot to add one of two primers. The number of molecules of single stranded DNA produced after 25 PCR cycle is. As you see, we have two primers in PCR, but when we perform PCR with only one primer, then this primer attach complementary to the one DNA strand, and this primer may not attach to other strand because other strand may not contain the sequence complementary to that of primer. And in process of extension, complementary nucleotides to the template molecule are added on 3' end of that primer, and only one new strand is synthesized. So here you can see after one PCR cycle we get one single stranded DNA molecule, and in second cycle also primer attach complementary to one strand and not attach to the other strand because other strand may not contain the complementary nucleotide. And after performing the second cycle we produce one more single stranded DNA molecule. So after two cycles we get total two single stranded DNA molecule. And likewise, when we perform 25 PCR cycle, then we will get 25 single stranded DNA molecule.